Hi guys, today we're looking at microscopy, core practical, AQR, OCR, edexcel. Yeah, I can't even speak properly. Yeah, well, this is a good chance of coming up in your biology paper 1. So let's get to the marks. First part, how to prepare a slide. Really, you can break this down into 3 things. We've got how to prepare a slide, how you're going to look at your slide using the microscope, and then how do you do your microscope drawings. So first off, let's get into it. First things first, what we want to do is place a drop of water on the slide. So we're going to pick ourselves up a nice easy mark by dropping some water onto our slide. So this is important. Why do we need that? This helps us float the specimen on there. So we've got our drop of water on the slide, super duper. That's really one there. What's the second point? Second point is using tweezers. I hope you all know what tweezers are, those sort of things you can pluck your eyebrows with. But using tweezers, place a thin specimen. Now you can say sample, if you want to say sample, thin specimen or thin sample on the slide, on water. Okay, so what we're talking about here is actually if you've cut a piece of onion or you've got some cheek cells or a piece of carrot, whatever it is that you actually want to look through, it needs to be thin. Light has to be able to get through it. If it's too fat, light won't get through it. So you're going to use tweezers and you're going to place the best tweezers in the world. Those are, you're going to place your specimen onto the water. Okay, so that's point number 2. Number 3, most specimens are colorless. So stain the specimen. Now, if you're in AQA, they specifically say using iodine, but OCR or edexcel, you can just say stain. And for most of the time, you can just say stain in AQA as well. So stain the specimen so that you can see it clearly. Stain the specimen to see structures clearly. Most of these things look colorless under the microscope or are transparent as they're very thin. And so we want to see it clearly. E.g. with... Take a nice orange because we all know iodine solution, orangey brown. Okay, and then the final part is to lower a cover slip onto the specimen. Now you might be thinking, what's a cover slip? It's that thin bit of glass you've probably used in class, and if you're not careful, it snaps in between your fingers. It's so thin and so fragile. In fact, if I put it on the desk here, anywhere here, I wouldn't be able to see it. Anyway, you place one of these cover slips onto the specimen. Some people like to say using a mounted needle, which is just a way of doing that. And then if we want, we could say press down gently. If you press down hard on top of this, you're actually going to squish the cells and smear them, and then they're going to empty their contents. You won't be able to see anything clearly. So press down gently to remove air bubbles. Air bubbles can basically get in the way of the details. So you have a finishing slide that looks, you've got your cover slip, and underneath the cover slip is your water, and underneath or floating on the water is your specimen. And then you're going to get your thumb and you're going to press down gently to remove air bubbles. Brilliant. That's how to prepare a slide, guys. Let's go through the key points there. What are the key points? Drop of water, 1 mark. Place the thin specimen on the water, 2 marks. Stain, 3 marks. Lower a cover slip, 4 marks. Press down gently to remove air bubbles. So remove air bubbles. So you can see that, yeah, is it important that you remember all of this? It would help, but there are key points within that that will score you marks on the practical. Let's get to the second part and hope that I've left enough space. So part number 2 over here is going to be using the microscope, and hopefully I am going to write a bit smaller here and leave myself enough room on the paper for you to see all the points rather than running out of room. So point 1, when we use the microscope. Okay, so we've got a microscope, and your microscope, this is going to be the quickest drawing of a microscope. You've got your stage. Okay, great, that will do. You've got some handles coming out of the microscope, and okay, there's kind of 2 handles coming out of the microscope. Let's label it in 2 different places. Okay, so using the microscope, first things first, select lowest power on objective lens. What's the objective lens? It's these 3 metal cylinders that are coming down on a light microscope. You want to select the lowest power to start with. You can always increase the power later. So select the lowest power. Number 2 is use coarse adjustment knob. So you're going to use the coarse adjustment knob to move the stage. Now, they're not expecting in these answers you to write an essay. So you essentially want to lower these objective lenses and or move the stage up and down till it meets the objective lens. And then you want to say, look through eyepiece. So every time you say one of these things, not only are you mentioning a keyword, but you're letting them know that you're aware of what the eyepiece does. So here is the eyepiece over here, and you're going to be looking through that and changing the objective lens by using the coarse adjustment knob over there. And then number 4, after we've looked through the eyepiece, we want to adjust the fine focus. So there's a second knob. 2 knobs on the microscope. Interesting. Right, you can adjust the fine focus to get a clear image. And then if you require to go into a bigger magnification, you can add select a new magnification on the objective lens. So if I'm going through how to use the microscope, now when these questions come up, like how to prepare a slide, how to use the microscope, they're generally separate. So you'll either get a 4 marker on how to prepare a slide or a 4 or 5 marker on how to use the microscope. You may get some calculations in there as well. So let's go through the key points. Select the lowest power on the objective lens. Use the coarse adjustment knob to move the stage. Look through the eyepiece and get it into rough focus. Then we're going to adjust the fine focus clear image. Okay, so it's about word connection. Fine focus is to get a clear image. The coarse adjustment knob is to move the stage and get you into rough focus as well. Okay, and if we're looking for the fine focus knob, it's over here. And if you're wondering what the stage is where you clip the specimen that you made in the first section. Right, there's a third part to these practicals, and this one actually hasn't been tested a lot, so it's worth going into, because maybe 2023, or maybe if you're not taking the exams, you're watching this video in 2024, maybe your year is the year that they go to this, which is how to draw your observations. Now, they haven't done how to draw your observations massively yet. There's been a 1 or 2 marker. There hasn't been a big 5, 6 marker on how to draw your observations. There has been how to use the microscope, and there has been how to prepare a slide, but we haven't yet had a big question on this, so maybe this will be the year. So let's pay attention. There are some clear points when you're doing a drawing. You must use a sharp pencil. And you might think, yeah, well, is that going to score me points in the exam? Yes, it will. If they ask you how to do the drawings, that could well score you points. And we want to say no broken lines. So you've got no lines that are broken. We have no crossing of lines. So you must not have any lines that cross. It's a lot of no's here. No shading or coloring. Okay, you mustn't start to get artistic if you're doing GCSE art and think you're going to draw a beautiful drawing. No, there's no coloring, no shading. And then label clearly. And then number 6, write the magnification. So loads of points to score here. Okay, guys, that's fairly clear there. Use a sharp pencil, no broken lines, no crossing of lines, no shading, label clearly, write the magnification underneath. So you always have to do that. Here is an example of a bad cell drawing. Oh, here's my nucleus. Let me shade it in because I'm such a good artist. No, we've just made a boo-boo. That ruined for the no shading. Right, number 2. Oh, I need to label this to score maximum marks. Nucleus, great stuff. Oh, let me label the cell membrane. Oh no, what have I done? I've crossed lines when I'm labeling. This is also going to lose you a mark. So really important that we have no cross lines and no coloring in, and also that we write our magnification. So underneath your drawing, write magnification and something like that, times 50. Right, guys. Very good chance for Bio Paper 1 that this is going to come up. This is core practical for AQA, OCR and edexcel Biology Paper 1. Remember, let's get to the marks.